Hello, everyone. Welcome to today's special colloquium. It's a great pleasure to have Professor William Bialek from Princeton today with us. So Bill Bialek is the John Archibald Wheeler and Battelle Professor in Physics, and he's also a member of the multidisciplinary Louis Seigler Institute for Integrative Genomics at Princeton University. He's a theoretical physicist who's broadly interested in phenomena of life in a very broad sense. And he's very well known for contributions to our understanding of how coding and computation works in the brain. So Bill has his bachelor's and PhD degrees in biophysics from California, spent a couple of years in Europe and also at the Institute for Theoretical Physics in Santa Barbara. And then he returned to Berkeley as a member in '86. And so from early 2000s, he has been a faculty member at Princeton University. More relevant to today's talk, he's author of a wonderful textbook, Biophysics, which many of us use as the primary textbook in our courses in biophysics and biological physics. Most recently, Bill has been recognized by the Max Delbruck Prize for Biological Physics, but also by a number of prizes like the Schwartz Prize for Theoretical and Computational Neuroscience, and he's also election to the US National Academy of Sciences. So Bill will tell us the context for this talk, but something that I was very keen to understand is the following, that the US National Academy of Sciences sort of recognized biological physics or physics of living systems in general as a proper branch of physics. And so it was a long document that Bill sort of chaired the committee for authoring the document. And there was a nice session where there were lots of discussions and suggestions invited from the worldwide community, and the document was released, and a huge document, very comprehensive one, and the offshoot of that is a nice way to develop biophysics as a proper branch of physics, and so its implications for both research and teaching in biophysics. So we thought it would be a great honor if we have Bill telling us about this general story about biophysics and also what its implications are in the broader context. So before I hand over, a couple of points. So usual Zoom etiquette applies. If you have questions, just put your question in the chat box. I will monitor them and I will ask you to unmute and ask your question. You can also raise your hand if you have a question to ask. So Bill will pause at some appropriate points and take a couple of questions. We can also have some more discussion towards the end of the talk. All right. Thanks once again, Bill. So the Zoom screen is yours.
Thanks, Vijay. It's a pleasure to do this. As Vijay said, the occasion of this is this report released by the U.S. National Academy of Sciences, and the link here, NAP stands for National Academies Press. So nap.edu, Physics of Life, easy to remember, is where you can find the report. Someday when all of the permissions for the figures and everything else are granted, there'll be an actual book, which will be downloadable for free in PDF, and you can also buy a physical copy of the book if you still indulge in such things. So let me tell you a little bit, as Vijay says, about context. The context is this decadal survey process. So I don't know how many of you know about this, but once every 10 years, the National Academies in the US undertake this review of physics as a whole. And the first time this was done was actually in 1966, which of course is not an even decade. So it took some time to get into that. That was the first attempt to survey the state of physics in the United States, and then it settled into a once a decade process. And of course, physics is a vast enterprise. And so, for example, in the cycle around 2010, the task of surveying all of physics was broken into 6 parts. There were committees set up to review astronomy and astrophysics, AMO, condensed matter physics, elementary particle physics, nuclear physics, and plasma physics. So what actually happens? What happens is officially the US funding agencies come to the Academy and say, would you please do this for us? They might need to be prompted in order to do that, but that's officially what's happening. The National Academies, as some of you may know, were founded. I don't know how the academies work in India, but in the US, the National Academy of Sciences, in addition to being an honorific society, is actually there to provide advice to the government. And so this is one of those examples where the government asks for our advice. What is produced is in response to that, to those requests. So it's not that you just get a bunch of condensed matter physicists in the room and say, tell us what you think about the state of condensed matter physics. No, the funding agencies have some specific set of questions they're asking. They're quite broad, but still there's a guide. An essential part of the process is getting community input, and I'll return to that in the context of our report. An important feature of these reports is that they're called consensus reports. So that means, for instance, that it is not the case that, you know, chapter 8 was written by Professor X and chapter 7 was written by Professor Y. The entire committee has to agree to everything. And so we speak with one voice by the end. It is the committee, as you'll see in a moment, is about a dozen people, and we have almost as many referees as we have committee members. So there's a very elaborate process for choosing the committee members, and for choosing the reviewers and so on, that's managed by the Academy. And I should say that different fields use this survey process differently. Most famously, I think, in astronomy and astrophysics, this is the process by which the priorities for large-scale projects are set for the country. So the fact that the James Webb Space Telescope was launched recently and produces these marvelous images goes back to a series of these decadal reports, which placed an instrument like that at the very top of the list of priorities. So this really does have its impact. So an important part of the audience for all of these reports is policymakers in the US government. And so the reason we're here is that in the 2020 cycle, for the first time, biological physics was given its own volume. So it was decided that there should be a review of biological physics or the physics of living systems, that we settled on. Since we were the first ones, we got to pick the catchiest title. So we just picked Physics of Life as the title for our report. So this is, I think, for our community, kind of a big deal. If you trace your way back, certainly in the 2010 cycle, if you look inside the condensed matter physics report, you'll find things about biological physics. If you go all the way back to the 1990s, then anything related to the physics of life was sort of in the back of some report about, you know, applied physics or something. By 2000, it had made its way into the front as something that might be interesting for the coming years. So the idea that now we're going to look at biological physics as one of the branches of physics alongside the more traditional ones really has been a kind of generational shift. And so that's part of what we mark here. So let me tell you who we are. Since it's a consensus report, I will sometimes speak in the plural. And it really is all of us speaking together. There's a wide range of colleagues who were who formed the committee that wrote the report. Theorists, experimentalists, people from a little bit more on the biological side, mostly people some people who came to biological physics late in their careers, after successful careers in other areas of physics. Some people who really made their reputation in biological physics, so quite a mix. Coordinating all of this and making it happen, especially during the pandemic, this is what we did with our pandemic. The very first meeting was in February of 2020, and of course it was also the last in-person meeting. So that was all held together by the marvelous staff at the National Academies, who really did extraordinary work, not just at a practical level, but also quite a lot of insight into how to say things that would actually have impact, which we hope will still be true. So we owe them a great debt. So again, if you want to find the report, just go to this website. This also contains a kind of guide to the report, which is quite well done, and again, entirely the work of the National Academy staff. We were asked a set of questions, as I mentioned, which were roughly, you won't be surprised, review the science in the field, tell us about the connections of the field to other areas of science and to technology and society, and then identify the challenges in realizing the potential of the field. So our report follows that outline. And now let me jump in and do something substantive. So one way of thinking about what biological physics is that like in the rest of physics, we see phenomena that strike us as being interesting, and we ask a certain kind of question that I think we all recognize as being physicist questions. So for example, if you think about the phenomena of life, and you try to describe what's happening in an organism or in a population of organisms in a single cell, what you find is that in order to describe that system, you need an enormous number of parameters. So if you want to think, for example, about how your brain works, well, you have neurons, individual cells, they're all connected to each other through synapses. And if you start trying to write down a model for the dynamics of that network of neurons, then that dynamics is controlled by the strengths of the connections, among other things, by the strengths of the connections among all the neurons, the strengths of synapses. And those parameters, there are lots of them. There are, in the central regions of your brain, every neuron makes thousands of connections. So in principle, if you want to know about a little corner of your brain that has a few thousand neurons, you have millions of parameters to keep track of. As a physicist, that makes you want to run and hide. So that's, we don't like models that have millions of parameters. And it's worse than that because, unlike the parameters that we use in more traditional, more better developed parts of physics, these are not, fundamental constants of the universe, right? They are literally plastic. They change with time. And there's a series of processes. So in the case of the brain, the most obvious way in which they change is through learning. But there are changes on shorter timescales inside cells, which are often called adaptation. And of course, there are changes on much longer timescales due to evolution. So somehow living systems, there's a view which has emerged from the biological physics community that you should think of any particular system that you look at as being an example that lives in this big parameter space. And over time, it kind of moves around. And you'd like to understand the dynamics of that movement and in some sense, what the point of that movement is. You expect, for instance, that if you're learning something, you're getting better at some task, and you'd like to understand how the dynamics relates to that improvement in your performance. And whether there's anything general to say about this. I mean, as a physicist, you don't want to think about all those parameters. Maybe there's something general to say about the dynamics by which the organism navigates this parameter space. And over the course of the last 20 years or so, there's been a tremendous influx of particularly theoretical physicists, but not only, into problems related to evolution. And understanding, for instance, that the dynamics of evolution can be cast as a statistical physics problem. And it's an interesting statistical physics problem because in some way, the way in which so first of all, one should note that in a population, there's a distribution of organisms, right? Not everybody has exactly the same genome, and therefore not everybody is equally good at solving the problems needed for survival and reproduction. And then that distribution has a dynamics, and in many simple contexts, you can see that dynamics is controlled by the tail of the distribution, not by the bulk, which is a sort of interesting, sort of captures the attention of statistical physicists immediately. Part of what statistical physicists have done here is to take things that have been talked about for a long time in the context of evolution and try and simplify them to the point where you can get full analytic control. And one of the things that's happened in that process is the discovery of regimes in which the trajectory of evolution is reasonably predictable. Evolution is often held up, I think, in part for political reasons in opposition to a creationist view as being something which is sort of random and contingent. And well, that's true in the sense that mutations occur randomly, but of course there are enormous selective pressures, particularly if you start thinking about the viruses that are trying to infect us and the way in which our immune system is responding to them. Those viruses are under enormous evolutionary pressure. And so as a result, aspects of the trajectory of their evolution are predictable. So this is a nice corner of statistical physics to play around in. But part of the reason that it gained so much traction was that while the theorists were exploring these models, experimentalists were producing tools, many of them coming out of the biological physics community, but not only, it's some merger of physics, chemistry, and biology, for rapid and efficient DNA sequencing. And so the result is that there's an explosion of data on the sequences, the genomes of viruses across the world, particularly those which are relevant for infecting human populations. So one of the consequences of all of this, so these theoretical ideas suddenly became relevant for thinking about analyzing these data. And conversely, the data provided you ways of testing your theoretical ideas and posing new theoretical questions. But this goes all the way, I mean, this seems like an interesting theoretical exercise, but it turns out that the ideas and indeed the people who came from the physics community to think about these problems of evolutionary dynamics found their way to the table where the WHO decides on next season's flu vaccine. And if you think about it, although evolution is often held up as being random and unpredictable, the idea that you're going to make a vaccine for the flu is to make a prediction, because you want to make a vaccine against the variant of the flu which appears next year, not last year. And so these ideas have found their way into that discussion in interesting and fascinating ways. And furthermore, in order to just deal with all this, the same community of people developed tools for collecting all of these. So there's the sort of experimental physics and chemistry part of actually doing the sequencing, but then there's the sort of data management problem, which, you know, large-scale data management is something the physics community has been involved in for, you know, before it was called big data. And so there are these tools that were developed by the biological physics community to track the evolutionary trajectories of viruses all across the world. And if you note the date on this reference, you'll see that it was conveniently just before COVID-19. And indeed, these tools are an important part of the global response to the pandemic. So this is a kind of snapshot of something that's happened in the biological physics community over the last generation, starting with curiosity about very theoretical questions, and leading and developing in this relatively short time to the point where it's had an impact on public health. This is a kind of beautiful example. So we should step back a little bit. This was just one vignette, and realize that physics and biology have been interacting with each other for a very long time. So if you go back to the 17th century, then, some of the first things that anybody saw under a microscope when microscopes were invented were single-celled organisms. So our ability to see the microscopic world, the first things that we saw were alive. In the 18th century, there were experiments on what was then referred to as animal electricity. And the names of some of these characters who did these experiments were Galvani and Volta, whose names you will recognize. We still have galvanometers, but they measure volts. There were debates about whether the electrical phenomena that you see in living organisms were special to life or a more general physical phenomenon. So this whole issue of where the phenomena of life fit into a larger picture of larger physicists' understanding of the world was already there in the 18th century. In the 19th century, so there are two major things, one of which is not illustrated here because we weren't sure what the right figure was. In the 19th century, as many of you know, the laws of thermodynamics were not considered established until you could balance the energy budget for the movement of living organisms. So you really had to convince yourself, in order to believe in the laws of thermodynamics, that energy was conserved even for living things. And that was crucial in both establishing thermodynamics and sort of starting to kill off the idea that there was some vital force that allowed living things to escape from the laws of physics. What is illustrated here is a different chapter of 19th century physics and biology. This is a drawing of the cochlea, the hearing organ buried deep in our inner ear. And as physicists understood acoustics and optics, it's fascinating to go back to this period and see that many of the practitioners did not see a boundary between acoustics and hearing and optics and vision. So, the giants of 19th century physics crossed that boundary quite routinely, and actually in many ways set an agenda for the exploration of vision and hearing well into the 20th century, and showed how very fundamental things that we were coming to understand about the physics of sound and light were directly relevant for how we perceive the world. And I think part of the reason for this fascination is that our eyes and ears are the instruments that we carry around to make observations on the world. And so the physics community found that idea very, very striking. And if you look into the literature of that period, you see that it wasn't only trying to understand how optics, the optics of the eye or the mechanics of the inner ear, the interest went all the way to understanding things about the inferences that we could draw, that our brains could draw, to use modern language, from the data that are collected with these physical instruments. So it really was a quite grand vision, as I say, set an agenda for a century to come. Okay, so moving up, by the time we get to the middle of the 20th century, the interactions between physics and biology are very strong. And of course, emblematic of that is the understanding of the structures of biological molecules, notably DNA, the famous photograph 51 taken by Rosalind Franklin, the double helical structure proposed theoretically by Watson and Crick. And it's not only that you could apply the tools of physics to learn something about the structure of biological molecules. First of all, those tools had to be extended enormously, so there's an interesting chapter there. But also this had an enormous impact on how we think about the phenomena of life. It is out of the idea of the double helical structure that there grows the idea that that DNA sequence encodes information about that is the repository of your genetic inheritance. That raises the question of what the code is and so on. And by the end of the 20th century, you have tools like functional magnetic resonance imaging that allow you to observe the human brain in action in a sort of non-invasive way. So you have this panoply raging over 4 centuries of interactions between physics and biology. And if you look at the history of successes, it's really extraordinary. And each of them is sort of foundational for generations of work that comes after. But an important point is that when you look at the history, these successful examples of interaction between physics and biology always get codified as being progress in biology. So it's an interesting question for the historians of science, for instance, how it is that in the middle of the 20th century, you have a relatively small community of physicists sitting in physics departments, writing the papers, which are absolutely foundational for what we now call molecular biology. And yet somehow when it was all over, it was molecular biology and not physics. So physics itself was relatively untouched by these dramatic developments. Now, somehow around the turn of this century, something happened. And the physics community started to appreciate that it wasn't just the application of known physics to problems outside of physics, but rather that by looking at the phenomena of life, you see challenges to our understanding of physics itself. And there are lots of interesting questions about what changed. As far as the committee could tell, there's no single eureka moment. There's many different events that happen that sort of push toward this realization. And so our task is not as historians to try to understand exactly what happened, but rather to assess where we are now and how we move forward. And because this is the first time that there's been a survey of this kind, in the introduction and overview, we do a little more work with the history than our colleagues do in other of these reports, but we're not historians. So our task is to discuss where we are now. So in trying to survey the science, what we tried to emphasize is that there's this incredible breadth of activity in physicists exploring the phenomena of life. And to organize it, we decided to think about the kinds of, about a set of rather large questions that the community has been addressing. And importantly, we've tried to identify these as being sort of physics questions, which are then illustrated by many different biological phenomena, as opposed to holding up a single biological system and saying, can you please explain to me how this particular thing works? What's striking, we think, is that there is this breadth of that activity and that it does organize, at least loosely, into these kinds of questions. So an example of the kind of question that the physics community has been interested in, the biological physics community has been interested in, is to try to define what problems it is that organisms need to solve in order to survive in the world. And in many cases, these are physics problems. You have to convert, or living organisms has to convert energy from one form to another. You have to move through the world. You have to control your movement. You have to sense your environment, despite the fact that signals are often weak and noisy. You have to build structures in space and time which are not on the natural scale of the component parts. And so just to give one example of this, in terms of energy conversion, almost all of the fuel for the active processes that happen inside our cells and the cells of all organisms, all eukaryotic organisms with a nucleus, come from the synthesis of a molecule called ATP by this very particular molecule that lives in cell or mitochondrial membranes. And this molecule is extraordinary. It is a rotary engine on the scale of nanometers. It sits in a membrane, and protons flow through the molecule. The cell works to produce a concentration difference of protons, or chemical potential difference, between inside and outside. So it's a battery. And the current from that battery flows through this molecule, which then rotates. And during the course of its rotation, it catalyzes these very particular chemical reactions that end up making a molecule called ATP, which is the sort of common cellular fuel, for example, for the contraction of our muscles. So to just give a glimpse of what the community has done, on the one hand, there's the experimental demonstration that this thing is actually rotating, which involves being able to manipulate the individual molecules on a nanometer scale and observe the rotation directly by very clever microscopy. And so here you can see it rotating, and you'll notice that it rotates in steps. So it's a discrete rotation. And there are, as you can see, three places where it pauses. And each of these transitions is associated with the synthesis of a single molecule. It's an extraordinary set of experiments. At the same time, this is an engine. There should be thermodynamics. So how do I understand thermodynamics on the nanometer scale with a single molecule where, of course, there's some reasonable probability that the molecule will rotate the wrong way because it's in the regime where its own Brownian motion is significant. And so there's been, as many of you know, a huge growth in the statistical physics and thermodynamics of small systems, and in non-equilibrium statistical physics more broadly. And the interaction between these kinds of experiments and that kind of theory has been extraordinarily productive. So here you have the phenomenon of life inspiring new physics experiments. I mean, being able to do this is really quite an impressive piece of experimental physics. And at the same time, providing inspiration for new theoretical ideas, and gradually the loop is closing. Another example of a big question that the field faces is that most of the phenomena that you're interested in are not like this. Most of them are emergent phenomena that involve interactions among large numbers of microscopic constituents. Now, in fact, if I wanted to take it seriously, I might point out that these molecules, although they're on the nanometer scale, they're of course very large as single molecules. And so their own structure emerges from the interaction of hundreds or thousands of their own amino acid constituents. And then this emergence is repeated scale after scale in the folding of chromosomes, in phase separation in the cell, which I'll talk about in just a moment, in the mechanics of the cell where large numbers of motor proteins and filaments interact, providing a paradigmatic example of active matter, and of course in networks of neurons in the brain and in the collective behavior of flocks of birds, swarms of insects, and so on, or also populations of bacteria as they interact in your gut or on the surface of your teeth, and many other examples. And actually really practical things like, you know, the surface of the rotary blades of a ship, which can be fouled by these bacterial colonies. So the figure here illustrates something which was discovered really only a little more than a decade ago, 15 years ago, which is that when you look under the microscope inside a cell, this is an in vitro system, or an artificial system that reproduces some of these phenomena. And if you look inside a cell, you see little regions which are bounded and carry out particular functions. And for many years, people thought that all of those were surrounded by a membrane that held the thing together. And what was discovered is that, in fact, many of these are condensed droplets of proteins and nucleic acids. And at the same time as that realization was happening, people went into the laboratory and made artificial examples where they could show that this happens just in a test tube under the microscope. And you see that these droplets are liquid, that they can merge and split in all the ways that condensed droplets should. And so this is a phenomenon whose observation has kind of swept through the biological physics community and through the larger community of cell biologists in the space really of just a decade. And it's becoming realized that there are an enormous number of functions of a living cell which take place in the context of these condensed droplets. And there's an effort to understand, how do cells control the phase behavior of their internal constituents? How do these condensed droplets influence function, and so on? Life depends on, for example, having enough energy in order to carry out your various functions and stay alive, but it also depends on having information. And information in living systems is represented on many different scales. There's information contained in DNA sequences. There's information contained in the concentrations of molecules, which is a sort of unusual example for physicists. We're used to the idea that information is transmitted by electrical signals, as it is in the brain, or by optical signals. But chemical signals are a different matter, and so there are lots of interesting conceptual questions there. Of course, information is represented in the brain, and we communicate with each other, and not just us, but also, you know, bacteria communicate with each other when they make a decision about infecting you, or how to live. You know, should they stick to a surface, or should they float freely through a solution? And there's been an enormous explosion of experimental tools and theoretical ideas about the sort of the physics of information in the context of living systems. And one beautiful example of this is that, as cells develop and as they change their environments and respond to new environments, they control their own internal state by synthesizing different proteins that are encoded in the genome. The intermediate in that synthesis is messenger RNA. And in order to interrogate the information that the cell has about the state of its environment and the control that it's going to exert on its own subsequent development, you'd like to measure the concentration of these messenger RNA molecules. And by a combination of combinatorial chemistry and super resolution localization microscopy, which is really sort of, we'll come back to this in a moment, it's now possible to reach inside a cell and essentially count every single messenger RNA molecule and have them labeled by their identity as, there are 17 messenger RNA molecules that were transcribed from gene number 172, okay? And you can do that for the whole list. And this is really remarkable. I mean, our experimental tools have gotten to the point where we can interrogate the state of a cell with single molecule resolution across nearly the full spectrum of molecular constituents, at least at the level of messenger RNA. It's really revolutionary. Finally, let me come back to this point I talked about of how living systems navigate parameter space. I gave you an example from evolutionary dynamics. Here's an example from the dynamics of learning. This cute little photo here is a finch, a songbird, wearing headphones. And the reason he's wearing headphones is so that you can control what he hears. So as he's singing, it turns out that, that these birds learn to sing. This is actually a Bengalese finch, which I hope is correctly named, but I don't know. What you may not realize is that they don't just learn once to sing. Like us, when we speak, in order to maintain our speaking voice, we have to constantly listen to what we're saying and fine tune. And so what that means is that you can perturb the song that is sung and drive it to do different things, and it will learn to adjust to, for example, a noisy environment. And there are beautiful experiments which really harken back to the kinds of things that were done in the 19th century, but now in a much more sophisticated way. And then quite deep theory saying that, your willingness to believe that you should change what you're doing is related to how noisy your actions are in the first place. So your susceptibility to perturbations and the dynamics of change should be related to the statistical structure of your own variability. And there is a principled way of doing this, which results in this sort of beautiful and largely parameter-free description of the dynamics of change during learning in relation to the underlying statistics of the bird song itself. And of course, this is an example where you can also chase this down to the level of understanding what individual neurons are doing. I think one of the things that the committee felt very strongly about, and that we hope comes across in the report, is that you have this, the physics of life runs from looking at individual molecules to looking at many individual molecules, to looking at collective behavior, to looking at the phenomena of learning. And it spans this incredible range, which hardly fits into any single biology department. Biology departments are often fragmented depending on which parts of this problem you're studying. But somehow for the physics community, there's some unity to all of this. And one way of keeping track of how this has happened is there's a Center for Educational Statistics, which is funded by the National Science Foundation, that keeps track of PhD recipients in the United States. And they do it in a fairly fine-grained way. They survey, for example, all the people who get their PhD in physics and ask them, which area of physics did you work in for your thesis? And of course, the biggest group is condensed matter physics. but they started counting biological physics as a separate field about 15 years ago. And over the course of those 15 years, it's shot up to the point where biological physics today is what elementary particle physics was 20 years ago. So, it's hard, one doesn't want to extrapolate these trajectories, they're noisy and so on. But I think the committee was incredibly surprised by this, because despite the fact that we all work in the field, we had this feeling that our community was sort of small and somehow nascent, that we didn't quite have critical mass. But that's not true. So we have arrived as a community as part of physics. These are people getting their PhDs in physics departments, PhDs in physics. And there is also a track from the same people for biophysics as a part of biology, and it's about the same number. So today, if you're interested in problems at the interface between physics and biology, you can either be a very physically minded biologist, or you can be a physicist who happens to be interested in the phenomena of life. And those are about equal. And the community of physicists interested in the phenomena of life is as large as much better established fields were just one generation ago, and growing fast. And you can also see this if you look at the number of talks that are given at the American Physical Society meeting and so on. So the first conclusion is that the field has emerged, and it sits alongside all these more traditional fields. And it's important that we in the physics community more broadly recognize this, and that we understand that, as with many areas of physics, there's a lot to be gained by interaction with other disciplines. So, if you do condensed matter physics, there are things that connect to engineering and to chemistry and to material science and so on. But that doesn't change the fact that you need a core of condensed matter physics in the physics department. And so similarly, the committee argues that the exploration of the physics of life needs to have a home in physics itself, even though there are wonderful connections to surrounding fields. And in thinking about these connections, the connection between biological physics and the rest of physics, you usually think, oh, the rest of physics gives us ideas, tools, real physics gives us theories, methods, and so on, and you get to apply them to biology. So things have changed as biological physics has emerged as a branch of physics in its own right. In many cases now, there are ideas in the core of physics, for example, in statistical mechanics, where the best experimental test is to do an experiment in a biological physics example. And so many of you are aware of the developments in non-equilibrium statistical mechanics that speak to fluctuation theorems and so on. And so the best experimental tests of these ideas are by single molecule manipulation of biological molecules. And the reason that you want to do something biological is that living systems have the ability to synthesize molecules with extraordinary precision. And so for those of us who grew up with the idea that physicists should stay away from the phenomena of life because they're messy, it's the kind of wonderful irony that the best experimental tests of deep physics ideas come from doing experiments with biological systems. Okay? And that's a product of this incredible interaction between theory and experiment in sort of taming the complexity of these living systems. It's of course also true that the biological physics community produces tools that can be exported to the biology community more generally, and then take on a life of their own, and people do all sorts of wonderful things with them. And one of the most obvious and general examples of this is breaking the diffraction limit in microscopy. So many of us grew up with the idea that the resolution of a light microscope is something of order the wavelength of light with some factor out in front, and that's that. And you sort of knew that wasn't quite right. There are ways of getting around it. But really, in this century, right, in the last 20 years, this has exploded. And as an example of this, here is an image of, which came from a movie, of a single bacterium where what the experimentalists have done is to track each of many different individual ribosomes, the factories that synthesize proteins, as they move around inside the cell. And what you can see is that you're tracking them with better than 50 nanometer resolution on timescales of tens of milliseconds. And these are very big molecules. So they themselves are, order 10 nanometers across. So you're almost tracking them with a resolution which is their actual size. And so you literally see the phenomena of life in action. And this is not a still picture, right? This is tracking microscopy. You see the phenomena of life live at the scale of individual molecules, 1 to 2 orders of magnitude beyond the nominal resolution of the light microscope. And of course, your ability to do this hinges entirely on your understanding of the physics of light. So this is really, if you want, a triumph of sort of AMO meets biological physics. And as I say, you can't buy that microscope, but you can buy super resolution microscopes. So if you're setting up a biology lab and you want to have a better view of what's going on inside a cell, these are now tools that are widely available. And again, that's happened very fast. I spent rather more time on the big questions because I thought that would be maybe more interesting for this audience. But of course, this issue of how what happens inside the biological physics community is connected to problems outside the biological physics community is it's an incredibly rich subject, which we explore in part 2 of the report. And there are things on the basic science side and on the sort of technology and society side. Let me remind you that the current revolution in artificial intelligence, for example, has its roots in efforts by the physics community to build models of the brain itself. And in fact, you can find in reviews of modern physics from circa 1960, articles about perceptron, which anticipate the architecture of modern deep networks. Of course, a lot of things had to happen in between, but these roots are very deep and very old. And so there's a incredible variety of subjects here, which really sort of strained the ability of the committee to reach across the whole thing. It's really quite remarkable to see how problems, very basic scientific questions about the physics of living systems have had an impact all the way into public health, the practice of medicine, and so on. So it's a very exciting time. There's lots of stuff happening. You can imagine that out of all this ferment, there's going to come a physicist's understanding of these beautiful phenomena. What's required in order for that to work? Well, we asked the community, and the community was very clear. We need to think about how we educate people. So the largest topic of input from the community, which came both from one in-person, so there was an in-person community input sort of town hall meeting at the Biophysical Society, and there was an online meeting organized by the American Physical Society, and there were paths for the community to provide written input through the website, through the project website. The strongest message was about education, and I think one way of the two observations are that when a new field emerges, it really gives you a chance to rethink how we teach, not just in service of that field, but in service of all the things that have happened since we thought about it last. And really, at least in the US, undergraduate physics education is now very far from how physicists think about the world. It really solidified, or solidified might be a polite word, maybe ossified, when before my generation was a student, right? We're still teaching in very much the same ways. And that's not good for our field of biological physics, but it's not good for lots of other fields either. And there are several ways of saying all this. for our field in particular, there's something special going on, which is that there's also an interest on the part of the biologists in producing people who have more quantitative approaches to biology itself. And somehow the problems of reshaping physics education in ways that would be good for physicists interested in the phenomena of life is not unrelated to the problem of reshaping biology education to give people a more quantitative approach to the same phenomena, although of course not exactly the same, because physicists ask different questions than biologists. And so it's a kind of complicated landscape. It's also true that the educational landscape is immensely variable. And so in particular, we said things about education in the US because there are dramatic differences when you step outside the US, just differences in structure. But even within the US, the majority of physics courses are taught not at universities that give PhDs and have research programs, but at community colleges. So when you talk about education, you have to be very careful to respect this enormous diversity of environments in which education is happening. That said, we tried to break our recommendations into groups. Some of them are things that can happen just inside physics departments. And the recommendation here is that there should be an effort to integrate biological physics or the physics of life into the mainstream physics curriculum at all levels. Today, you can get a first degree in physics from a first-rate institution and not even realize that biological physics exists as a field. And it's difficult to imagine that could happen with condensed matter physics or elementary particle physics or astrophysics, even if you never take a specialized condensed matter physics course. Examples from condensed matter physics are woven in to our teaching of statistical mechanics and quantum mechanics and so on. And the report unpacks this recommendation by looking at the ways in which examples from the physics of living systems could similarly be integrated into the physics curriculum. Not as a separate thing, not as an application, but just as examples of fundamental physics principles in a different context than we usually give. Some things you can't do only in the physics department because they involve this interaction between physics and biology. And their universities, at least in the US, are spending substantial resources on programs in quantitative biology, biological engineering. And it's important that the physics community get its share of those resources and engage with those efforts because the physics brings a unique perspective to these problems. And finally, this is a very US specific recommendation. On the biological side, most PhD programs in the biological sciences and the biomedical sciences in the US get direct support at an institutional level for their PhD students coming from the National Institutes of Health. And there is no analog of that in physics at all in the US. And so in particular, right, from our perspective in biological physics, we see this contrast. And so there's a specific recommendation that federal funding agencies should establish grant programs for direct institutional support. And so there's an interesting discussion to be had about the models for support of PhD students in different places. How I would be interested to learn how it's done in India. This is something that, again, I'm going to go quickly through this and wrap up with our recommendations. I'm speaking to an audience in India. A large number of fantastic physicists in the US got traced the beginnings of their education to India and came to the US afterwards. I think you're all aware that for many years, the US had a kind of privileged position on the world stage as an extremely attractive destination for people who have the ambition of a scientific career. To be blunt, and I think the report is blunt, we've given this advantage away by a variety of actions, some more disturbing than others, both matters of policy and matters of rhetoric. And it is the strong recommendation of the committee that this needs to be fixed. We need to repair our relationship with the world. by emphasizing the open exchange of people and ideas. And the situation is serious enough that we cannot do this by rhetoric or visa policy alone, but we actually need to put money behind it. And so many of the programs for support of PhD students exclude international students for a variety of historical reasons. And so we need to return to an era in which exchange of scientists among countries is viewed as a positive form of diplomacy, which it was, well, certainly in my youth. But we've come very far from that, and we need to go back. That's the recommendation of the committee. I think an issue which is universal, although of course we phrase it in the US context, is that the physics of life is not about a particular problem. It's not about building a fusion reactor. It's not about finding what lies beyond the standard model. It's about an approach to a wide range of problems, which are still maturing. And the unification is not the things that we're studying. They all happen to be alive, but that's a pretty broad category. But rather the kinds of questions that we're asking and the kinds of answers that we're looking for. And in contrast, with the exception of one program in the National Science Foundation, which is quite small, all of the sources of funding for our field are fragmented along various axes. And it is the conclusion of the committee that this fragmentation is actually bad for the field, and it's bad even for the missions of the agencies that are supporting the field. And so all of the agencies need to find a way of supporting the field in its broad, coherent form as it exists. So essentially, for a generation, the field has succeeded by adapting to the funding landscape. And it is our recommendation that it is now time for the funding landscape to adapt to the field as it is actually practiced. I think what I'd like to do is to stop here, because the remainder of the recommendations are very specific to the situation that we find in the US. So let me go to the end, also since it's a bit late, and summarize by saying that what this report does is to kind of celebrate the emergence of biological physics as a field of physics. It is something that all of us felt very excited to be a part of. It marks a very special moment for our community. And this report itself has been very much a community activity. Although there's a dozen or so of us who were responsible for writing the report, it draws crucially on community input. And it's our hope that most people in the community will find something of themselves in the excitement that we try to communicate about the field and about the challenges that we face. And the conclusion is that we did our part in digesting the community input, and it's time for the community again, both to ask questions about where we are and to do things. So we make recommendations which will not happen just because we said it was a good idea. It will take effort by the community to turn these recommendations into reality. And so we're excited to watch as that happens. So let me stop there and thank you for your attention. And I'd be happy to chat about whatever.
Thanks, Bill. Thanks very much. It was a wonderful and fabulous talk. A couple of questions in the chat box, and people can also raise their hands and ask questions. So there's one question from Vakesh. How would you differentiate between a biologist, a physicist, and a biophysicist?
Yeah. So I think. So let me answer. Let me. We tried not to be prescriptive. There's clearly a continuum of activity, right? From things which are very biologically motivated to things which are physics motivated. I think that our definition of biological physics is you're asking questions that are recognizably in this traditional style of physics, and you're addressing them to the real phenomena of life. So there used to be things which were referred to as biologically inspired physics, where you looked at the phenomena of life and you abstracted a few things away and you studied those, either by building synthetic systems in the laboratory or by studying models. And this is beautiful work. I think one of the things that's been exciting is that in many cases, there's been a sort of circling back. So, the problem of active matter, as you well know, and many other people in the audience, I suspect, has its roots in trying to build models for flocks and swarms, and produce beautiful theoretical physics. And then people started to make real measurements on real flocks and swarms. And it turns out that real flocks and swarms have extraordinarily regular and precise and reproducible behavior that is in a different universality class than in the models that people wrote down. And there's still an active search for, how do I find the models that describe, what is the underlying theory that describes the real system? So I think that this is an example of this kind of change from thinking about, you can look at the world and see something that inspires you, but you don't want to engage too directly, to really saying, no, the real physics of life challenges our understanding of physics itself. There are new things out there that we didn't think of. And so that's I think where the field sits. And it's not useful to put a hard cutoff, because at some moment you might have said about that work, just to give an example, well, yeah, but it's not really connected to actual flocks of birds, so I don't care. Or maybe the fact that it's not connected to real flocks of birds it makes me happy, because then I don't have to think about the complexities of real birds. But that wall has been removed, and that's exciting. I hope that helped answer the question.
I hope to. Rajesh has a question.
Hi, Bill. Great talk. Very eye-opening. Thanks very much. So I had a bit of a long shot question, which is, I was very impressed with what you said about the precision tests of non-equilibrium statistical mechanics. And I was just wondering whether, do you see a potential in the next decade or whatever for for systems and biology being able to shed light on foundational questions of quantum mechanics about macroscopic coherence, or even quantum information? Just please interpret it broadly, in any sense.
So, like many physicists, I had my period of being fascinated by the question of whether quantum mechanics was directly relevant to biology or to life. My own forays in that direction, the first one, we were just wrong. The second one was more modest but more successful, but it was at a more molecular level. I think that there. So, for instance, something that's sort of somehow less dramatic, but might be actually quite important to how our lives work, is that the sort of whole understanding of where the border between quantum and classical behavior is for thinking about chemical reactions has undergone quite a lot of change. And it's now understood, for example, that almost all of the reactions that involve transferring a hydrogen atom in enzymes, the proton is actually tunneling. And so that's an example where, tunneling is not as dramatic as coherence, but it is true that the community had it wrong, and it was something we thought we understood. And so there are also examples where that you may have heard about in the process of photosynthesis, both in energy transfer on the way, capturing the energy of the photon and sort of propagating it toward the place where the chemistry happens, and then in the very first chemical steps, all of these things are so fast that the decay time for coherence and the time for the transitions are kind of on the same order of magnitude. And there's a lot of discussion about what are the experimental signatures of that? How does it matter for function and so on? And I anticipate that something will get resolved there. There is some flow of this back into thinking about what's been called bath engineering, right? So that by somehow the usual argument is, if you want to do fun quantum things, you should slow down the process of destroying coherence. But these examples have inspired people to think about slightly more subtle things, where you say, well, the process, coherence is destroyed by interacting with a bath, and the detailed spectral properties of that bath matter in some cases for how things proceed. I don't know that any of this is revolutionary yet, right? Or very crisp. I think part of the problem is that the phenomena in living systems where quantum mechanics, or let's say quantum coherence, is unambiguously relevant, are still on rather small scales of space and time. And so they're experimentally hard to get at.
But you mentioned the ATP thing. I mean, that was also so
right.
different.
So that's an example, right? So things keep progressing, and our experimental friends keep surprising us.
So it's probably not something that
that's not where I would. I think, like with many things about the foundations of quantum mechanics more generally, right? We sort of know that investing zero in that is a bad idea. But on the other hand, it's not clear what's ripe at the moment. And so I think clearly it's sort of pleasing as a physicist to see these quantum mechanical effects being important. But when it'll become crisp enough that you say, oh gosh, I realized there was something about quantum mechanics I didn't understand that is driven by this, that's a little harder. Although this issue about the crossover with proton tunneling, I think, was kind of fun. Shows you that it's possible.
Great, thanks.
Spenta.
Hello, Bill.
Nice, Spenta.
Very instructive, very inspiring, and important talk. I have one question about something you mentioned in the beginning of your talk, that this report was requested by some agency of the United States government. Can you a little bit detail that?
Yes,
process?
sorry, I didn't mean to be cryptic. The reports in different areas of physics tend to be sponsored by different agencies. In some cases, many agencies cooperating. In our case, it was the National Science Foundation that came to us. And that's not coincidental. I would say the National Science Foundation, the report reaches the conclusion that the National Science Foundation is the only agency that has a program that sort of views the field as a coherent whole and as part of physics. So there is a biological physics program, or physics of living systems program, inside the physics division of the National Science Foundation. It's a small program. It extends itself by partnering with other programs. A remarkable discovery for everybody involved in the committee is that an enormous amount of money flows into the biological physics community from the National Institutes of Health. We defined the biological, there's a problem of identifying, which part of the money the National Institutes of Health is spending is on biological physics. There is no biological physics program. So we did the simple thing of saying, well, if they send the money to a physics department, it's probably biological physics. And that's approximately right when you look in detail. The National Institutes of Health gives out money through things that are called study sections, which are basically grant review panels. And there are something like 300 of these. And the physics community has received money through 75 different ones. So this is what we mean by fragmentation. And you can understand then why the National Institutes of Health did not come to us and say, you should be worried about this, because what is the this that we're talking about? The National Science Foundation had a view of what this might be, and we were charged with trying to articulate that. Now the challenge is to spread that message to more agencies. I hope that helps.
I know, just asking the question a bit more. So from what you said, it seems that there was a cell in the National Science Foundation of physicists or scientists who requested the report. But
Yes.
Where does the government come in here?
So that's a matter of some creativity. I mean, there's always an issue with these reports. As I say, the National Academies are constantly doing this, not only reviewing the state of physics, but worried about transportation and healthcare, all sorts of stuff.
Yes.
How you get the people who spend money to listen is complicated. So, for example, before the report was officially released, among the people that we brief were the Office of Science and Technology Policy, which sits inside the White House, and staff members who work with people in the Congress and the Senate on the relevant committees that control expenditures in the sciences. And one of the things we heard actually at the very beginning of the process from these congressional staff people is that they love these reports and have them on their shelves with little Post-it notes in them, so that when something comes up which is relevant to what's going on, when something comes up in Congress that's relevant, for which one of the reports is relevant, they can pull it out and show it to the members of the relevant committee. So that's grounds for optimism, but it's not a plan. And so that's part of what the Academy staff is spending its time on, is trying to get the report into the hands of the people who actually make decisions. And that's also a community activity. So several of us will be speaking at a forum sponsored by the American Physical Society Division of Biological Physics. And of course, so we as authors in writing the report, our advocacy ends when we write the report. And that's very important for reasons that I'm sure you understand, for maintaining our objectivity. And in particular, these reports are viewed by people in decision-making positions as being fair and authoritative. And so that means that there's some boundary of advocacy that we cannot step over to maintain that position. However, the community, right, the American Physical Society can advocate. And so we'll see how that develops. It's just been a couple of months now.
Thank you very much.
I think I need to give shorter answers because there's lots of questions.
You should do that. I'm losing track of keeping.
Okay, let me look through the chat and just try to rattle off a few things. Is there buy-in from other physics subdisciplines for our recommendations about teaching of physics? We're going to find out pretty soon, because there are some discussions that will be happening about physics teaching. I think a lot of people view the interactions with our field as being relevant only to the teaching of physics for pre-medical students, and we're sort of fighting against that. I don't think there's dramatic pushback with good examples. I think that we will get traction. Although it's this is a new point, and it remains to be seen how it works. What books and courses do you recommend for a graduate level physics student to make a research career in neuroscience? We, as a matter of the report, we decided not to make a list of resources for fear of leaving things out. It's also true that several people on the committee are the authors of textbooks, myself included. So we wanted to be a bit careful. So if you want to write me a note about my personal taste, I'm happy to do that. We don't have an official position about what to recommend, but there has been a huge impact of the physics community on modern systems and computational and theoretical neuroscience. And so there are a lot of resources out there. So please feel free to send me an email. Visa restrictions. The committee was very strong about the easing visa restrictions. To particularly, we were concerned about its interactions with education, and we're happy to report that even before the report came out, some of the things that were among the worst possibilities were avoided, largely through the advocacy of the American Physical Society. Your point about international conferences is interesting. I think on the whole, so the position of the committee is that we need to return to a positive view of international scientific exchange as a form of diplomacy. This is, by the way, a view which the United States managed to maintain even during the Cold War. So it would seem like we ought to be able to maintain it today. We're pretty explicit about some of these things which involve technical issues about policy that I don't want to go into here, very US specific. Should you not have an international conference because you can't invite all of the people that you want to invite and be sure that they'll get visas? That doesn't seem like a positive step to me, but that's a personal opinion. It seems like what you want to do is, if you want to implement these things, the scientific community in the US has to keep up pressure on the government to push for visa policies being what they should be. And I would say that we're not alone in making these recommendations. There's been a whole stream, as part of this decadal survey process, there's been a series of these reports coming out, and they're all coming down on the same side of this issue. And when we briefed the Office of Science and Technology Policy, the first words out of the mouth of one of the senior advisors was, "You should know that this administration is very different than the last one." And that we're very much in favor of these things. So I think there are grounds for optimism. Do we want to, let's see. I don't want to miss the people who have their hands raised, but let me see if I can make some progress on the ones in the chat. Ideas and theories and how cells perform so fast and accurate. Yes, so there's a great deal of discussion in the report of examples where you see extraordinary precision in the functioning of biological systems. And this is clearly something that has captured the imagination of the biological physics community more broadly. And there are certainly ideas. This is something I personally have worked on, and so I want to be careful to draw the line between what the report says and what I think. If you dig into the report, you'll see a lot about this. Let me leave it at that. For those of us who work with elementary and junior high school age kids, could you give a few examples of what's exciting? I'm not going to do that, not because I don't think it's important, but because the report decided, in writing the report, we decided that pre-college education was beyond our remit. It's incredibly important, and we do give a lot of examples of how the phenomena of life can work their way into the core physics curriculum. And what you might think about is whether some of those examples can be sort of scaled back to the point where they are accessible to pre-college students. Again, one of the things that we say, that we recommend, is that there is a need, actually I can't remember whether this is a conclusion or a recommendation, there's a need for collecting resources. The American Journal of Physics used to have, certainly continues to have, these sort of resource collections, and it would be good to have some in our field. Are there more questions?
People have raised hands.
Yes, let's do the raised hands. We can come back to the chat.
Madan?
Yeah, thanks, Bill. Very, very nice talk, and I'm really impressed by the U.S. scientific community, where they can actually make these decadal assessments and proposals for the future. I wish we had such things in India, and maybe people like Spenta and all can propose that in our context.
Parenthetically,
yeah.
when I tell my biologist colleagues that I was involved in doing this, some of them said, "Oh, that's interesting. Maybe we should do that in biology." So it's not. I don't think it's something unique about American culture. It's something about the U.S. physics community and its interactions with the academy and the government, and I don't know, has some history.
Yeah, excellent. Yeah. So I was just going to quote from Stanislav Ulam, who said that that ask not what physics can do for biology, but ask what biology can do for physics. And I thought that fits in with what you
absolutely yes.
Yeah. So the question, I had two questions, and I don't know if I'll have the chance to ask these both, but let me start with the first one. So, one of so this has to do with education and especially education in the Indian context. And, the examples that you quoted of the coming together of physics and biology were all sort of inspired by deep phenomena in biology. And so unless a student has an awe for those phenomena, you're not going to excite him or her. Now, it turns out that the sort of best students of physics in India are more enamored by frameworks, mathematical structure, and so on. And so, how would you then go about teaching it? Could you? I guess one would like to now build a sort of a collection of structures and mathematical frameworks that are unique to biology, which don't exist in other fields of physics, and so that would mean a different kind of curriculum. And so, have you thought about those?
Let me make a personal aside. The problem that you're referring to, which is what motivates students, I'll remind you which institution I teach at. Perhaps the typical PhD student in physics at Princeton is not so different than the ones you're describing. I think this answer is different at different levels. I think that for instance, this is an example we give in the report. Sorry, actually, let me back up a little bit. One problem is that the way we teach physics leaves untouched the suspicion that experiments on living systems are irreducibly messy and there's nothing you can do about it. And in fact, I would say one of the great triumphs of this last generation is to tame many of these systems to the point where you can present them to a physics audience without needing to know enormous amounts of details and produce, show data that looks like a physics experiment, right? And so I think part of the thing is that, you might say, well, it's hard to get people excited if they don't know something about the biology. But, how do you explain to somebody that superconductivity is interesting? Well, I have to tell you that there's this thing that, you measure most materials have a resistance, and then I cool the thing down and the resistance goes to zero, right? And you should be surprised by that. And then why that turns out to be so fundamental, that you should really be worried about the Meissner effect and not the zero resistance, and all that takes longer, right? But somehow, as one of my colleagues once said, when you see the liquid helium climbing up the side of the dewar, right, in the fountain effect, I don't need to explain to you, I don't need to convince you that this needs an explanation.
Right.
So I think there are phenomena in the living world, I have my own favorites, so I don't want to pick those that have that kind of flavor to them. In terms of integrating into the curriculum, I think there's lots of opportunities. Every statistical mechanics course talks about the random flight polymer as an example of entropic elasticity, right? In some version of this. The best experiment on that is picking up a single molecule of DNA and pulling on it. It's a beautiful experiment. Also, it's a physics experiment, right? The way you do it is with magnetic traps or optical tweezers. Optical tweezers being the subject of a physics Nobel Prize for. And similarly, when we teach quantum mechanics, we talk about energy levels and atomic absorption spectra and so on. But look around you, you don't see a lot of spectral lines. If you look at the stars, you do. But if you look at your shirt, or your skin, the colors don't come from spectral lines of atoms, right? They come from these very broad absorption spectra of big complicated molecules. And so there's an interesting physics problem just to understand why they're so broad, right? They're not broad because of lifetime broadening, right? It's because of coupling among many degrees of freedom. So we stop, there's many places where we stop short of doing things which I think are just beautiful examples of deep physics ideas that are immediately relevant to sort of everyday phenomena of life. We just don't do it. We don't point out that if you take classical mechanics and add a little bit of friction, then you break time reversal invariance. But if you're sitting in a fluid and you go all the way to the limit of low Reynolds number, where the fluid has no inertia, you restore time reversal invariance. And as a result, if you go through the same sequence of movements, your progress through the world doesn't depend on the times in which you do it. It only depends on the sequence. And that means that if you don't explicitly break time reversal invariance in your sequence of movements, you won't get anywhere. And this has tremendous implications for how microorganisms move around. It's also true that if you explore the world of low Reynolds number hydrodynamics, right, that you can plausibly think of this as objects deforming themselves, right? But of course, the natural ways of describing deformations have a kind of gauge freedom to them, right? And so there are these hidden gauge structures in low Reynolds number mechanics. There's all sorts of beautiful stuff to engage people. And so I think it's just a matter of figuring out how to do it.
Yeah. So if I may, can I ask one more question?
Okay, short one, quick one.
Yeah, quick one. Yeah. So in as you said, that the exercises that you went through is the physics community trying to announcing that biological physics is now a respectable discipline of its own and has its own structure and so on. But ultimately, I mean, a desirable feature would be for biological physicists to be able to work together with biologists and together solve important problems. But unfortunately,
so I think the position of the committee is in response to our history.
Yeah. Okay.
So I think historically, the idea that physicists would go collaborate with biologists, the biologists would be the arbiters of what was interesting. They would phrase the problems, and physicists would provide tools, whether those tools were experimental or theoretical. I think that has been the mainstream view. And of course, biology is a very large enterprise, right, and provides a happy home for large numbers of physicists. The thing that was missing was to articulate that there was another part to it. But in the same way that condensed matter physics is not only interesting because we have semiconductor devices, right? There's, it's really important, and it's a major part of why the field grew to be as big as it is, but that's not the only reason we do it. And so we wanted to articulate the other part.
Okay.
And so that's where we put the emphasis. It's not, and actually, okay, so part of it being a consensus report is that once it's written, you sort of don't go back to revisit the arguments. But I think the report also explains that because there's this continuum of activity, it can be difficult to draw circles, right? And to some extent that's subjective. And as I think I answered in relation to another question, to Vijay's question, that when we were talking about active matter versus real flocks and swarms, if you put a boundary, the chances you're going to have to move it or erase it are actually pretty large. And that's part of what the scientific progress is, right? That, things that once upon a time were viewed as physicists going into biology and doing something useful for the biologists. So you look back and you realize, oh no, wait, there's actually a physics problem there that I don't understand. Maybe once you understand that, it becomes part of biology, and that's fine, right? It's the emphasis is rather that we will not make progress we will not realize our intellectual ambitions. We don't nurture the part that sort of physics community has a kind of unique responsibility for.
Right. Yeah. Thanks.
Thanks, Will. Pramod? Pramod?
Yeah, can you hear me?
Yeah.
Yes.
I can switch on my. Okay, somehow my video cannot be switched on, but that's okay. Yes, I am Pramod from Ramar Research Institute. See, my question is a little bit from the biology perspective. Of course, the main recommendations which you suggested was how we can include biophysics into physics education and so on. But it seemed also interesting to think what changes could be made into biology education. And one of the things which comes to my mind, and I would like your opinion on that, is that there are significant differences in how people approach problems in these two communities. And biologists often, for example, do not like the idea of simplifying a problem at the expense of physiological relevance. And physicists often would like to simplify the problem to the maximum extent and then build it up from there. Another area may be abstract, ability for abstract thinking and so on. So apart from the requirement of mathematics and so on, I was wondering whether, what your thoughts are on, if we were to sort of advise biologists as to what changes.
Right. Let me make a couple of observations. First of all, 20 years ago, there was a report, another one of these reports from the Academy called Bio 2010. It was written in 2000 and was looking ahead a decade.
Mm-hmm.
And it was specifically about what we as a community needed to do in order to produce a generation of biologists who would have more quantitative and even theoretically oriented approaches to the subject, to realize the promise of emerging technologies, eventually even, getting to things like personalized medicine and so on. What the committee chose to do was to reinforce many of those messages and emphasize sort of the particular role played by physics in addressing these problems. An example is the relationship between theory and experiment in physics being somewhat unique, right? So there's quite a bit of the chapter about education, which is addressed to what you might think of as interdisciplinary education. But it quite specifically was not our job to tell the biologists how to teach biology. And so there's a line there where we're talking about the things that, where the physics community should be engaged in the efforts to build a more quantitative biology. And the fact of the matter is that at many institutions, the physicists are engaged in those efforts. In fact, in some places, they're leading those efforts, but the efforts are nonetheless perceived by the university administration as being efforts in biology. And so the funds flow through the biology departments. And so there's a problem for university administrators of figuring out how to support. I mean, they like to talk about doing interdisciplinary things, but really the fundamental business of the university of teaching students, the dollars sort of follow the students around, and being sure that for instance, if you want to have programs in quantitative biology, you're providing the support for what happens in the physics department that is in the service of those efforts, and more strongly that physics, along with mathematics and parts of engineering and so on, have a voice in the intellectual structure of those programs. That's a problem that has to be solved university-wide. And so we talk about those issues again in a very US context, building on the things that some very far-sighted biologists and a few physicists who were on the committee said 20 years ago.
So we have in India, we have a set of institutes where they have or they offer much more flexible curriculum for both for biologists and physicists, ICERs, for example.
Yes.
But often when you think about teaching some basic physics to biology, one is talking about certain physics topics. But it's also important to realize that there is a difference in the way you approach problems. So that is what I'm
so there's quite a bit of emphasis in the report on and you may have heard this even when we were talking about funding, but it's of course also true about education, and there's crosstalk between those discussions in the report, that the issue is often about intellectual style, right? It's the kind of question we ask and the kind of answer that we expect to arrive at, more than some particular technical thing that we're doing. Of course, both physics and biology are filled with lots of technical things, and they're different technical things, but that difference in attitude is very important. It's difference in
absolutely, yeah.
Well, a couple of questions.
Teaching that is not simple. It's not even simple to say, in what, where are we transmitting our sense of physics culture to our physics students?
Mm-hmm.
We do it somewhat implicitly, right?
Yeah, absolutely.
You're supposed to deduce that there's this style of thinking.
Yeah.
And I think that if we want to export that style of thinking to a broader community, it's a bigger market, if you want to use capitalist language, then we need to be a little bit more explicit about what that is. And that's a challenge. That's a challenge for us in the physics.
Absolutely, and I guess it's onus is on physicists to sort of think how we can sort of I do not know.
Yeah, I think it's also true that one of the things about the Bio 2010 report and our building on that is about dialogue at the beginning regarding intellectual goals, rather than saying, I mean, it doesn't work if I, as a physicist who doesn't know much about biology, said, in order to prepare physicists to think about the problems of the biological world, they should learn this, 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 and this. Chances they get it right aren't very high. So similarly, if the biologists sort of say, I want to pluck this, 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 and this from the physics curriculum, the chances that those are the right things aren't very high. And so there really needs to be a more fundamental dialogue. And we talk about the challenge of doing that. In particular, I don't know how it works in India, but in the US, you have some teaching assignment, and that's what you do. And the university administration would, of course, love to see innovation in teaching, but they're often not so willing to pay for it. So it is very seldom that your teaching assignment is to meet once a week with your colleagues from another department and figure out what you're going to teach the next time you get together, right? But that's what it needs to be if you want to have something genuinely new. And so that's really a question about resources.
Thank you very much.
There's also a backup of questions. Shall I just take a quick look through the chat?
Yeah.
So we were planning to see,
are there more examples? Yeah, you were around the elementary, junior high school kids.
Right. So we did that one,
Yes.
or at least I tried. More examples. The report's filled with examples. We really tried to be very broad. So let me just refer you to the report. On a relative scale, experiments in soft matter physics, biological physics, are probably less expensive compared to some traditional disciplines. Can that motivate funding-constrained developing nations, notably India, to take up this area in a higher priority? One could even go further and point out, for instance, that a lot of what happens in conventional biology is very focused on a limited number of organisms as examples. And places in the world where there's greater biodiversity can provide new inspiration. And there are actually important examples of this in the history of biology and medicine itself, but it could be broader. So, in particular, as our tools, experimental tools for exploring the phenomena of life have become more general purpose, the investment in a small number of model organisms is perhaps less necessary. And so I do think that there are opportunities in the developing world, although in some ways, right, I mean, yeah, India is very developed particularly in physics compared to many places. Somehow you're managing. So the question of what India's priorities should be are up to India. My goodness, the last thing I would want is for a report to be read as telling other countries in some sort of colonial fashion what they should be doing. That would be a disaster. So I think asking what are the opportunities. Let me make a broader. When you have a relatively new growing field, institutions and presumably nations, but let's think about it institution by institution, right? That are not, if you want to compete at the thing that everybody already understands is important, then you compete with the people who have the most resources. If you want to do something that not everybody has quite figured out is important yet, right? Then there are big areas where you can lead, even if you're not traditionally one of the people who has the most resources. And that's true for the growth. There are physics departments in the US who have tried to focus in areas which are not elementary particle physics and condensed matter physics. And I think it's true on a worldwide scale. So I think that's a very interesting discussion, but we certainly don't want to make any recommendations. Does the origin of life fit in? Certainly. How much priority do you think to give to such research as opposed to physics of life already existing? We were explicitly asked not to make priorities. And so we didn't, and we were relieved not to do that. We admire greatly our friends in astrophysics who managed to do this priority setting exercise, but we were very relieved not to have to do it. Do you see biological physics and physical biology differently? Are they as different as Indian Americans and American Indians? Either this is a marvelous pun, or you perhaps don't know that American Indian is used to refer to the indigenous people of the continent. So either way, do I see these as differently? So I think I've said before that the committee emphasized that there's a continuum of activity from biology with a little bit of physics sprinkled in to physics with a little bit of biology sprinkled in, and there's some core in the middle, which is the thing that we're talking about. But we tried very consciously to have a kind of big tent, as we say in American politics. Is interdisciplinary science the future? Sure. I would remind you that you don't have interdisciplinary science without disciplines. So the example that I think we may have actually used in the report, I don't remember, but I've certainly used it in discussions with many people, is there is something sad about the fact that there are novels I would like to read that are in languages I don't understand. The solution to this problem is not to insist that everybody write in one language, right? So the borders between disciplines reflect deep cultural history. And some of those are constraining, but remember that the borders between disciplines are not carved in stone, they move. The thing that happens inside physics departments today is dramatically different than what it was one generation back. And so I think crossing disciplinary boundaries is very important, but I think the thing which we focused on was the moving of disciplinary boundaries. So in some sense, you could say that what our report marks is taking the border between physics and biology and moving it so that some part of the living world belongs to the physics community in some intellectual sense. And obviously you can't do that if you don't walk up to the border and see what's there, right? So in that sense, interdisciplinarity is essential, but it's the disciplines that support this. What do you think of the fact that there are 3 academies of sciences in India? I don't know the structure. What I would tell you is that in the United States, there is the National Academy of Sciences, the National Academy of Medicine, and the National Academy of Engineering. And they each, the Academy of Sciences is the oldest, that's 150 years old. And the others were formed later. It wasn't originally the National Academy of Medicine, it was the Institute of Medicine. And it's only relatively recently that this has all been consolidated. It actually led to important difficulties, such as the fact that it looked as if when the academies spoke on issues related to medicine, it seemed to be the Institute of Medicine that was speaking. And when the academies spoke on issues related to, let's say, the natural sciences or engineering, it seemed to be the National Research Council that was speaking. And so, of course, the border between natural science, engineering, and medicine, if you're asking for advice for the government, it's not useful to divide them up that way. And so bringing this all together was very important. There are also other academies, but these were the ones that were established by the government for the purpose of providing advice to the government. Okay, thank you for the kind words. Officially used statistical models of biology that life keeps on evolving. How does the system? So look, this is a kind of, I mean, the real answer to this question is technical. I would say that if you look through the report, you'll find many examples where ideas that have a probabilistic or statistical character are used in thinking about the phenomena of life. And these range from things where it's sort of obvious physics, why. So, you know, if you think about the problem of protein folding, it's clearly a statistical mechanics problem because you have a molecule that, you know, adopts some more or less equilibrium structure that's non-trivial. And so if I tell you the sequence, the amino acid sequence, and ask you how it folds up, that's sort of obviously a statistical physics problem. On the other hand, you also think about the evolution of the sequence itself. And there, the notion that there's a statistical mechanics is less obvious, but many people in the community have worked to construct it. So I think that and I would also remind everyone that just because you use statistical models doesn't mean that you don't make deterministic predictions. Mm-hmm. So, you know, in the n goes to infinity limit, many things come true with probability 1. And so this whole interplay between statistical and deterministic ideas is very important in biological physics as a whole. How can physics be introduced to biomedical engineering students? Well, in the spectrum of things that we've talked about, biomedical engineering is relatively easy because, at least in the US, they would be sitting in an engineering school, and the curriculum set by the engineering school has has components from physics. And so that's not the biggest problem that we face. It might be structured differently in India. What's the relationship between biological physics, bioinformatics, theoretical neuroscience? So this is again the problem of drawing boundaries. There are people who came from the biological physics community who, you know, understood that certain kinds of string matching problems that were phrased by the computer scientists as versions of the issues that come up in the analysis of DNA sequences could be recast as statistical mechanics problems. There's been we talk about the analysis of DNA sequences in regulatory regions using a combination of statistical mechanics and information theoretic ideas. There's a whole generation of theoretical ideas in neuroscience that really trace to foundational papers written in the physics community. And although we scrupulously avoid credit assignment in the report, I think you can trace many of those things if you look through if you look through the relevant sections. I think there are parts. I mean, part of the ambition of the report was specifically to reclaim for the physics community many things that were done by physicists in a decidedly physics style. And I think sometimes the physics community has the confusion that if you do something that's related to biology, and the biologists actually think that that should be incorporated in part of the mainstream of biology, then it stops being physics. And there's a very, I mean, in addition to, you know, whether this makes you uncomfortable or not, which is a kind of subjective question, it has a very practical problem, which is that you might then think that there isn't a physics of that part of biology, except that that's where it came from, right? And so if you want to keep having, I have no problem with the idea that physicists do things that are connected to biology, they become part of the mainstream of biology, and they take on a life of their own. But if you want that to keep happening, if you think that's a source of creative and impactful ideas, then you need to nurture the thing that happens in the physics community. And that's the thing that we've tried to draw a circle around.
Okay, so that brings us to the end of all the questions. So Bill, thanks very, very much on behalf of everybody at ICTS. It was a wonderful talk. We thoroughly enjoyed it. I've also been getting a long stream of direct messages saying thanks for organizing this, and people really enjoyed, and you've given them enough material to think about teaching biophysics. Thanks once again. It was wonderful.
My pleasure.
And thanks everyone. Thank you very much for joining.